Well, welcome to the GW Epilepsy Grand Round. Today we have Professor James McNamara from Duke. Professor McNamara is professor of neuroscience at the departments of neurobiology, neurology, and pharmacology and cancer biology at Duke. He earned his MD from the University of Michigan at Ann Arbor and spent his career investigating the um, uh, cellular and molecular mechanisms of epileptogenesis, a process by which the normal brain becomes epileptic. A lot of patients with epilepsy experience an initial inciting episode, whether it's status epilepticus or otherwise, and then there's a latency phase before the development of epilepsy. And Dr. McNamara's research provides some evidence for a novel strategy targeting receptor tyrosine kinase signaling as a potential therapeutic promise for prevention and disease modification of temporal lobe epilepsy in humans. This is one of the current research interests that has led to a very famous, I have to say, paper in Neuron a few years ago, and gained a lot of visibility in the media as well. But his contributions to epilepsy research are too numerous to list in this introduction. But Dr. McNamara has earned a number of awards and honors, including being a member of the National Academy of Medicine, and also Javits Neuroscience Investigator Award from NIH, Freedom to Discover Award in Neuroscience from Bristol Myers Squibb, American Epilepsy Society Research Recognition Award, among numerous others. So without further ado, Professor McNamara.
Good morning, Mohammed. Thank you for the kind introduction. I'm delighted to be here this morning. Excuse me. And I am very much looking forward to sharing this story with you. And I will say at the outset, I'd be very interested in feedback, ideas on specific clinical trials in which we might test this idea, in different forms of epilepsy. So let me tee it up that way. And so can you see my screen?
Yes, it's perfect.
Perfect. Okay, good. So disclosures at the front end, Neurop Incorporated, I co-founded that company. LVM Biosciences is a company that I founded targeting the current the therapies that are the topic of this presentation today. I'm also on the Scientific Advisory Board of Pappas Ventures. So take whatever I say with a grain of salt. So this is the outline of the presentation today. We'll talk about temporal lobe epilepsy and evidence implicating a causal role of status epilepticus, receptor tyrosine kinase signaling, specifically TrkB, and how we might go from molecules to medicine, and finally, specific therapeutic considerations. So, as Mohammed mentioned in his introduction, a number of people that have temporal lobe epilepsy, it arises as a consequence of some insult. And work that Jackie French, among others, had done, retrospective studies recognize that many adults with severe temporal lobe epilepsy had experienced an episode of prolonged seizures in childhood. John Henniger's and others had done a longitudinal study which revealed that children with prolonged seizures were at increased risk. Work done at Duke by Daryl Lewis and Kevin Van Landingham revealed hippocampal injury after status epilepticus, which inspired the FEBSTAT study, a prospective study of children with status epilepticus, revealing acute injury that evolves to hippocampal atrophy and in turn temporal lobe epilepsy. The fact that you can induce, take an otherwise normal mouse, rat, cat, dog, or non-human primate and induce status epilepticus, stop it. And at some latency thereafter, the animals become epileptic, has led to the idea that status epilepticus is one cause that contribute to the development of temporal lobe epilepsy. Let me shift gears now and talk about receptor tyrosine kinase signaling, and specifically the brain-derived neurotrophic factor or BDNF receptor TrkB. So here is a schematic of this receptor, and I will explain this receptor is both a receptor and an enzyme, and it's by its enzymatic activity that it signals. So it lives in the membrane as a monomer, and when its cognate ligand, brain-derived neurotrophic factor, is secreted as a dimer, a homodimer, it triggers the dimerization and a conformational change in the cytoplasmic or intracellular region, which increases its affinity for ATP. And this enzymatic activity, it catalyzes the phosphorylation of tyrosines and specific residues in the cytoplasmic domain of the receptor. And those phosphorylated amino acids have increased affinity for an adapter protein like SHIC or the enzyme phospholipase C gamma 1. And it binds to them, phosphorylates them, and that activates signaling pathways, the RAS-MAP kinase AKT pathway and phospholipase C gamma 1. So why are we talking about TrkB signaling? And the answer started with an observation by Chris Gall approximately now 25 years ago, that here status epilepticus produced a massive increase in the expression of this gene in an adult animal. So this is in situ hybridization of a coronal section of a rat brain. And you can see here the hippocampus, what is white there are silver grains for messenger RNA for BDNF. 6 hours on the right, 6 hours after a seizure, you see this massive increase in the content of BDNF, demonstrating that in an adult that was otherwise normal, this abnormal activity, status epilepticus, produces a massive increase in its expression. Well, that triggered a number of studies which demonstrated that engineering a mouse with too much BDNF makes these mice epileptic. Furthermore, Kakaya and colleagues demonstrated in a kindled model that BDNF mice in which one allele was eliminated, they had impaired epileptogenesis. And work from our lab demonstrated that a conditional deletion of TrkB prevented epileptogenesis altogether in that model. And it's the only such genetic or pharmacologic perturbation that has such powerful effects. Now, it's essential, however, to align your animal model with the clinical condition you seek to study. And while it seemed plausible that status epilepticus contributed to temporal lobe epilepsy, and there was abundant evidence implicating BDNF and TrkB in different models of epilepsy, the question was status epilepticus, in a model in which status epilepticus induces temporal lobe epilepsy, is TrkB required? This is not a trivial question to answer, far easily posed than answered. Here is the model that we used to begin to address this question. And it was work based upon people like Vic Nadler and Ben Aree in the 80s, and then adapted to a mouse by David Henshaw in Dublin. And here in an adult mouse, stereotaxically implant an injection cannula overlying the amygdala, an electrode over the contralateral hippocampus so we can record EEG. Allow the animal to recover. A week later, inject a little bit of canic acid into that amygdala, and it triggers status epilepticus recorded both behaviorally and electrically. We terminated, just like the patient in the emergency room, with benzodiazepines after 40 minutes of status. And 3 to 7 days later, these animals developed spontaneous recurrent seizures. And these seizures will persist for the life of the animal in that model, the way we were using it. And so we will do video and EEG recording, just like you have a patient in the monitoring unit in one of our hospitals. So to test the idea, our hypothesis was that transiently inhibiting tract B after status epilepticus would prevent the development of temporal lobe epilepsy. And to do this experiment, and this is work that Mohammed was referring to earlier, we did this in these mice that were genetically modified, and it rendered them susceptible. Tract B could be inhibited with this small molecule, 1-NMPP1. We induced status epilepticus, and following terminating it, treated the animals with either vehicle or 1-NMPP1 for 2 weeks, and then we stopped it. And the half-life of this drug is very short. And then studied them a month later. Now, this on the vertical axis, the y-axis here, each one of these is an animal, and it's the number of seizures recorded during 2 weeks. Now, remember, these animals had had tract B kinase inhibited for 2 weeks following the status. Then we put them on vacation and came back later. And this animal had 28 seizures in 2 weeks. This animal just had 2 seizures, but these are controls, and both the wild type, the wild type animal, whether it was treated with vehicle or 1-NMPP1, each animal had many seizures. And the mutant animals also had many seizures, but the question was, if you treated those mutant animals with 1-NMPP1, and in those animals alone, the 1-NMPP1 inhibits tract B signaling, what happens to the seizures in those animals? And that's what happened. We virtually prevented the development of epilepsy. 7 of 8 animals had 0 seizures, and 1 of the animals had a single seizure. I should point out that all of these recordings were reviewed by 2 different individuals who were blinded as to the treatment of the animals. So what did we learn? We learned that it's possible, this is the first instance in which a treatment was introduced after the status epilepticus, just like a patient comes to the emergency room. You give them the benzodiazepines, and then you would intervene and treat them with this drug. So it's possible to intervene transiently following the status and prevent the epilepsy. We also studied a comorbidity, anxiety-like disorder. And it also provided proof of concept of tract B signaling as a drug for a target for drug development. Well, the good news from that was that we could initiate inhibition of tract B following status epilepticus and continue it for just 2 weeks and prevent epilepsy. That was the good news. But there was some bad news. And you're a clinician, you're sitting there asking, all right, so what's the downside of treating this animal? And the answer to that is, in work that we published in the last several months, we demonstrated that transiently inhibiting tract B, if you study them 24 hours after that status epilepticus, that there's a striking increase in the amount of cell death that's caused by the status. So treating a normal animal with this inhibitor has no effect. But status kills some neurons. And if in the presence of status, you inhibit this tract B, you kill many more neurons. So we reasoned that that could be a problem when we spoke to a parent in the emergency room, or we spoke with the FDA. So the question is, how might we get around that? And what Yang Wang, a very talented young scientist in our laboratory, raised the the idea is that perhaps distinct signaling pathways downstream of tract B could mediate the desirable, namely the neuroprotective effects of tract B signaling, and the undesirable, the epileptogenic effects of tract B. So what we know is that global inhibition of tract B in the context of status epilepticus promotes death of neurons. So what we wanted to do was to identify the dominant disease signaling pathway. And what we hypothesized was this Schick pathway from residue 515 was mediating the survival of the neurons. And if we inhibited that, we get more cell death. And we reasoned that perhaps phospholipase C gamma 1, which had been implicated in long-term potentiation, was the epileptogenic pathway. So this young man, Bin Gu, who is a graduate student in pharmacology, is completing postdoctoral studies with a colleague at UNC, Ben Philpott. Terrific young kid on the job market. And if you're interested in somebody that's going to be a real winner, I'd urge you to interview him. So what Ben did as a graduate student is we designed a phospholipase to inhibit this interaction. So it was known, the motif of tract B that binds to the SH2 domain of PLC gamma 1, those amino acids were known. And we made a peptide, and we took the 14 terminal amino acids of tract B, and then we fused them to a TAT sequence from HIV that enables a peptide to cross membranes and cross the blood-brain barrier. And the idea was that the PLC pathway was the one that was producing the epilepsy. And so the idea was that if we could block that pathway, in other words, we give the animals this peptide that would bind the PLC gamma 1, prevent it from interacting with tract B, prevent PLC activation, and prevent the epilepsy. So that was the idea. So how did we begin to test this? And I would point out that at the beginning, the status epilepticus produces a striking increase in the activation of tract B and the signaling pathway. And we could measure its activation of phospholipase C gamma 1 using Western blots. So here, if you induce status, and just look at this column here, these are Western blots. And this just take it on faith for right now that when this is very dark, it reflects activation of PLC gamma 1. So if a PBS treated animal, there is basal activation, but very little. But status epilepticus produced a striking increase in this without changing the protein itself or actin. And here it is quantified on the right. You see that striking increase induced by status epilepticus. So, but the question was, could we inhibit that with our peptide? And so what Ben did is induce the status, and well, actually, in this particular experiment, he just gave it to a normal animal. And you can see marked in or about a 30 or 40% reduction of phospholipase C activation without any change in these other signaling pathways read out as AKT or ERK. So this was both effective and selective. And so Ben went on to test this following status. So in this experiment, he induced the status, stopped it, and then treated the animals. And once again, you can see in the PBS treated animal, very little activation of PLC, an increase here, and then a dose-dependent decrease of phospholipase C gamma 1. So this dose response curve enabled us to figure out what dose to treat the animal to try to prevent the epilepsy. And the question was then, does treatment with PY816 initiated following status prevent the temporal lobe epilepsy? So we infused the KN8, status occurred, stopped it with diazepam, stopped it with lorazepam, and we treated the animals with a peptide once a day, each day for 3 days. And what happened to the seizures? And here in a scrambled control animal, this animal, for example, had 10 seizures in the first 2 weeks following status, a control animal. And there was a striking reduction in the animals that had got PLC, PY816. But the more relevant question was, what happened a month later? And so we resumed video EEG recording a month later. And once again, each one of these animals had multiple seizures. This guy had 38 seizures in 2 weeks, and this guy had about 8. But what happened if we had treated with this peptide for just 3 days after the status? And the answer was a dramatic reduction. There was a 90% reduction in the numbers of these seizures. Excuse me. So this peptide disrupts the binding of TrkB with PLC gamma 1 in vivo. I didn't show you that. It selectively inhibits the activation of that signaling pathway. And treatment with it for just 3 days following status inhibits the development of temporal lobe epilepsy. And importantly, what I didn't show you was it retained the neuroprotective effects of TrkB signaling were maintained intact. So we had our cake and ate it too. Now I'll point out that the sequence of that peptide and the sequence of TrkB is identical in a mouse, a rat, and a human. So this very drug, this very biologic, can be used as a drug in humans. And in full disclosure, we're working on advancing this peptide to the clinic. Now, I want to shift gears here a moment and talk to the putative role of isolated seizures in progression of temporal lobe epilepsy. So William Gowers, who was a very shrewd neurologist in London in the late 1800s, from observing his patients, he raised the idea that seizures beget seizures. In other words, what he was looking at his patients that had had, for example, a traumatic brain injury, a stroke, some insult that caused the epilepsy. But what he noticed was that once they had a seizure, the likelihood of getting another one was increased, and that the seizure seemed to the disease worsened over time. And he suspected that it was seizures themselves that were doing it. And this is exactly what this is what we're talking about. This sort of electrical activity here recorded from a CA1 pyramidal cell in work that done by Steve Turnells and Ray Dingledine many years ago. This idea was reinforced or supported by studies in animal models by Graham Goddard. The idea is quite simple, that whether the initial cause of the epilepsy might be genetic, like a mutation of SCN1A, it could be status epilepticus, it could be traumatic brain injury, it could be a stroke. But the idea is once they produce the epilepsy, that then the seizures themselves could contribute to worsening. And I think that this happens in some patients. It doesn't happen in every patient. But the idea is, if this is true, that then the seizure-induced worsening could be a process common to epilepsy of diverse causes. Well, if that's all true, the question is, what's the molecular mechanism by which this isolated seizure produces a worsening of the epilepsy? Because if you understood that, then you could have a target with which to intervene pharmacologically. So we hypothesized, it wouldn't be surprising to you at this juncture, hopefully, that the activation of TrkB PLC gamma 1 signaling induced by an isolated seizure is the molecular mechanism by which seizures beget seizures. And we thought that for a couple of reasons. First of all, a seizure, an isolated seizure, produces a transient increase of BDNF and activation of TrkB and PLC gamma 1 signaling for approximately 2 to 3 days. And a single seizure does. And furthermore, as we've just shown you, transient inhibition of this signaling pathway after status epilepticus, a far worse insult, prevented epilepsy and anxiety-like disorder. So how could we begin to test this idea? And I'd like to introduce to you Kamish Krishnamurthy, pictured here with his wife Krutika. And Kamish completed these studies during his MD PhD studies in my laboratory. And he is now a first-year resident in neurology at UCSF. Succeeding famously is what I would expect of him, nothing less. So Kamish used this kindling model of epilepsy to test this idea. And I mentioned this was discovered by Graham Goddard. And the way it was done in these particular experiments by Kamish, an adult mouse was implanted with an electrode in the amygdala. The animal was allowed to recover. A tiny stimulus administered, and it produces no change in behavior or EEG. But then that same stimulus by day 4 produces an electrographic seizure that remains localized. By day 7, that same stimulus produces a longer one, and the animal has what looks like a temporal lobe seizure in a human. By day 10, the stimulus produces a temporal lobe seizure, but then a secondarily generalized tonic-clonic seizure. And that persists for the life, that enhanced sensitivity persists for the life of the animal. But we use this model, Kamish used this model, because here is an example of seizures alone promoting worsening of the epilepsy. And I would point out that if instead of stopping at 10 stimulations, you gave the animal 70 such stimulations, the animal will develop spontaneous recurrent seizures, and many of these animals develop SUDEP and die. But the other part of the reason for using this model is we had temporal control of the occurrence of the seizures, and that temporal control was essential for us to test our hypothesis that the seizure itself, the activation of this TrkB signaling pathway by the seizure, was producing the worsening. So here was the design. So remember, our hypothesis is that inhibiting the seizure-induced signaling pathway, signaling of TrkB PLC gamma 1 will prevent the increased duration and severity of a subsequent seizure. So in these experiments, Kamish stimulated the animals until there were 3 consecutive seizures of class 4 or more. So here is stimulation number on the x-axis, and here on the y-axis is the severity of the epilepsy. And Kamish was studying them after approximately 10 to 15 such stimulations noted by this arrow. Remember, if we stimulated those animals 50 times, they start to develop spontaneous recurrent seizures that become refractory, and ultimately SUDEP is common. So in these particular experiments, Kamish, quote, kindled the animal, 3 consecutive seizures, and then he gave them a week's vacation, and then carefully figured out the current required to activate a seizure. And then following that, he treated the animal with either a scrambled peptide control or PY816 for just 2 days. And then he let the animal, survive for another 1 to 2 weeks. In this instance, it was 1 week. In the experiments I'm going to show you, it was 2 weeks. So the first question is, what happens in the absence of treatment with like PY816 the worsening. And what you can see here shown as the duration of electrographic seizure. The duration of seizure number 1 here, we're going to arbitrarily, each animal serving as its own control, we'll call that 100%. And if you studied them 2 weeks later, the duration of that seizure had increased by approximately, in these particular experiments, by approximately 50%. So the question was, if he intervened following this for just 2 days, could he prevent this worsening? That was the hypothesis. And so what he did is, and he's done this both with a chemical genetic approach, and he has done this with this peptide. And I'm going to show you the experiments with the peptide. And so the idea is, if he treated the animal following the seizure, just like you might do for one of our patients, they have the seizure, and then you treat them with this peptide. And the idea is it binds to the PLC-AMA1, scavenges it, and the idea is it would prevent the progression, prevent the activation of this signaling pathway and the progression. And indeed, this is what he found. And I'm going to show you that the transient treatment with this peptide initiated, I emphasize, after the seizure, not before the seizure, after the seizure, it reduced the duration of the subsequent seizure. So here is the experimental paradigm. Kindled, seizure number 1, we're going to have the duration of it, treat for 2 days, give the animal a 2-week vacation here. And what happened? So here in seizure number 1, the duration was basically 100%. In this experiment, seizure number 2 was about a 20 or 30% increase. So we expected to prevent that with PY816. And to our surprise, the duration of that seizure decreased by almost 50%. So it not only prevented the increase, but indeed there was a regression, a reduction. So this was electrical seizure activity. What happened to behavioral seizure activity? And same experiment, and now we have a different readout of seizure intensity. And this is the total duration of the seizure read out as tonic-clonic seizure activity and post-ictal suppression. And in the control animals, again, there was about a 20 to 30% increase. And what happened if you treat them transiently with PY816 for 2 days and then studied them 2 weeks later? And the answer was a striking reduction, about a 70% reduction of the duration of that seizure. Now, this was unexpected. What we expected was that we would prevent that increase. It's not that we prevented that reduction. So that led us to ask, well, what if we just forget about this seizure? We just kindle the animals, give them a week's vacation, and then treat them with the peptide or the scrambled control for a couple of days, and then study them again 2 weeks later. Could we induce this regression? And the answer is no. So here is the experiment that the duration of that final kindled seizure, 6 days vacation, no seizure here, simply treat for 2 days, and then test the animal again. And what happened? And the answer is they both increased about 20%, and there was no difference between the scrambled control and the PY816. So to summarize, a single seizure caused increased duration and severity, behavioral severity of the subsequent seizure, additional evidence of seizures begetting seizures. Brief treatment with this inhibitor of TrkB signaling alone did not modify the duration or the severity of that subsequent seizure. But treatment with that inhibitor initiated after the seizure attenuated the severity of the subsequent seizure. So to reiterate here in this schematic, this was the kindled animal. This is where he started. What Kamish expected was that if he treated them with this peptide, that he would prevent this worsening going from here to here. But what he found quite unexpectedly was that if he induced the seizure, then treated him with the peptide, he actually got regression. They moved back down along this line. So unexpectedly, our findings revealed that a seizure, we interpret this, is it's activating some signaling pathways that reverse the epileptogenesis. And these beneficial consequences are revealed only if an inhibitor of TrkB signaling is administered following a seizure. So paradoxically, a seizure actually provides an opportunity for the brain to heal itself of this disorder if, following that seizure, the signaling pathway activated by TrkB and PLC gamma 1 is selectively inhibited. So our model is that the seizures do beget seizures, and that worsening is mediated by TrkB, PLC, gamma 1. And it's both the seizures and it's some of these comorbidities that I really haven't discussed today. But simultaneously, we think that seizure is activating a homeostatic pathway that has beneficial effects. We don't know the identity of this signaling pathway that is in turn inhibiting the epileptogenesis, if you will. So we would submit that Gowers was absolutely right, Gowers and Goddard after him. Clearly, seizures do beget seizures, but seizures can also beget a homeostatic pathway, or, I'd be interested in how you might suggest that we say this, seizures also beget no seizures. If following the seizure, a PLC gamma 1, TrkB PLC gamma 1 inhibitor is given. And I should emphasize at this point, in data that I haven't shown you, Kamish also used that chemical genetic model and demonstrated that inhibition of TrkB kinase itself had the same effects as this PLC inhibitor. So in the last few minutes, I'd like to turn to some therapeutic considerations. And I would urge you to put your thinking caps on and provide me with some of your ideas as to how we might exploit these findings to benefit humans with temporal lobe epilepsy. So for sure, one might be that intervening with a TrkB PLC gamma 1 inhibitor following status epilepticus may prevent the development of temporal lobe epilepsy. That's a challenging clinical trial. Prevention trials are always more challenging than disease modification. Long-term readout, aligning patients, stratifying them for risk of developing the disorder, long-term follow-up. It's absolutely doable, important to do, will be done, but a tough clinical trial to begin with. But with respect to disease modification, in my view, a key question is, at which stages of the disease are these beneficial consequences of a seizure effective? We've shown it early in the course of epileptogenesis. The question is, does that signaling pathway promote this progression at later stages? And among the questions is, might treatment with a PLC gamma 1 inhibitor briefly following an isolated seizure reduce the frequency of seizures and or transform temporal lobe epilepsy from being medically refractory to medically responsive? And if we were going to do that, so here the idea would be potentially patients teed up for a temporal lobectomy. And might we use them to begin to design a clinical trial in which we used this inhibitor initially to see if we could induce a remission or if we could transform medically refractory to medically responsive. And might there be other considerations for a design of a clinical trial? So in closing, I want to thank you for your attention. I want to thank the National Institutes of Health, specifically NINDS, for support of our research program spanning 4 decades. And most importantly, I want to thank the American taxpayers whose hard-earned wages support NINDS and their support of this line of investigation. So I thank you for your attention in this unusual format. It's challenging to present under these circumstances. And I hope that I've been able to convey some of this information, and I look forward to any questions that you might have. Thank you.
Thank you so much, Dr. McNamara, for the outstanding talk. I would like to tell the viewers that the code, the CME code to earn CME is 14102. Again, 14102. And if this number is texted to the following mobile number, then you will earn automatically the CME. The mobile number is 202-509-9968. And again, the code is 14102. Thank you, Dr. McNamara. We have a couple of questions, and I'm encouraging viewers on the web also to type in questions, if any, in the live webcast chat box on our website. But I will start with the ones I have here. The question is, let's see here. Does the peptide prevent, excuse me, does the peptide prevent neuronal cell death? In animals who showed significant reduction of seizures after treatment, was this paralleled by histology that showed less neuronal cell death?
Okay, so first of all, we've done these experiments following status. Okay, so the answer to that is a little bit complicated. Okay, when we study animals 24 hours following status epilepticus, you would normally get cell death. Let's say you'd kill 100 neurons in the hippocampus. Mm-hmm. If you induce status epilepticus and inhibit tract B signaling, and specifically globally, and you get and kill the animal 24 hours later, you'll get 200 cells dying. Now, however, if you induce status epilepticus and you treat the animal with our peptide, you'll only have 100 cells dying. So it's retained the protective effects of other tract B signaling pathways. Okay, so that's acutely following status epilepticus. If you, now we're going to ask a different question. You induce status epilepticus and you treat the animal with a peptide for 3 days. And then you study this animal, say a month or 6 weeks later, and you look at the amount of cell death. There will be less cell death in the animals in which you had treated them with the peptide. And I think that is probably due to the prevention, the intact signaling pathways acutely following status. But I also think that there's ongoing cell death in these animals, perhaps related to flurries of seizures. So that the absence of those seizures in the peptide-treated animals is also probably producing some neuroprotective effects. So a long-winded and a bit complicated answer, but I think that I answered your question.
Yes, thank you. The other question may be related on the behavioral level. Canic acid is known to result in significant memory and behavioral issues. You kind of alluded to anxiety, but how about memory problems and whether they appear to be partially mitigated in treated animals?
Okay, so a really good question, Mohammed. And we've not tested memory. We did, as I mentioned, test anxiety-like disorder with a light-dark test. And I'd be happy to elaborate on that. But in this model, this particular model, the canic acid is not administered systemically, but rather into the right amygdala. And the cell death in this model is virtually restricted to the ipsilateral hippocampus. That, with respect to hippocampus, in any event, the contralateral hippocampus is virtually spared of cell death. And these mouse models of memory, the vast majority of them, well, we're unaware of any, like you could do Morris water maze or radial arm maze, etc. But with damage to a single hippocampus, they don't, it's not detectable. So obviously, if this were a human, we'd be looking at visual spatial memory in the non-dominant temporal lobe or verbal memory, but we can't do that in a mouse. Or we can't, maybe somebody else can.
Yeah. Thank you. I think this is not a question, but a comment. A lot of epileptologists who have patients who undergo epilepsy surgery and become seizure-free while still on medications. Sometimes the patient stops their anti-seizure medications, right, and they have a seizure.
Yep.
And then even when they resume taking the medicines, the seizures persist. Suggest, again, this is probably not studied with high-class evidence. This is an observation from clinical epileptologists. The thing is, they stop the medicine, they have a seizure, and then even when they resume the medicine, the seizures persist. And this may provide clinical evidence for the fact that seizures beget seizures. So it's like as I said, there's no question here. It's just like a comment.
Yep. I agree with you completely. And that's why when I was practicing, I would retain patients on low dose of a single anti-seizure drug following the temporal lobectomy to ward off that possibility, because I have seen patients like that. Anybody that works in the field has seen patients like that. And I agree with you that it's anecdotal, but it's like, it leads to that approach, namely keeping them on a single anticonvulsant.
Yes. Another question is, was the treatment in animals intravenous or intrathecal? And whether intravenous treatment in humans may lead to immune responses?
Yeah, all excellent questions. So when we did the TrkB kinase inhibitor, that one in PP1, we gave that to them orally and intraperitoneally. But specifically with respect to the peptide, in the experiments with the status epilepticus in the prevention of the epilepsy, that was given intravenously for 3 days. In the experiments that I described in the Kindling model, that was given intraperitoneally. Okay. Now, with respect to the immune issue, I agree that that is an important consideration. And obviously, one concern could be developing an autoimmune, neutralizing antibodies that could prevent the effectiveness in time if one had to give it repeatedly. or in principle, you could induce an autoimmune disease. We've not seen any evidence of that. I can tell you what we are doing is we are intent on developing the peptide as a therapeutic, and similar approaches were just read out with another peptide, NA1. It was reported in Lancet about 3 months ago. This uncouples an NMDA receptor subunit from a scaffolding protein, and it was used in a phase 3 clinical trial for stroke following mechanical thrombectomy. So I do think that it's possible, that it is feasible, but I am concerned about that. And we are in parallel trying to develop a small molecule that would do the same thing as the peptide.
All right, thank you. I think there's one more comment and one last question here. The comment is, there was a recent study about individuals who have seizure clusters in the epilepsy monitoring unit. And the authors concluded that the last seizure in the cluster was the longest in duration, which probably one remembers that study because you mentioned that seizures can indeed prevent seizures if the peptide is given, or if the BDNF pathway is somehow not active. And I think this comment aims to investigate whether the longest seizure has potentially more likelihood of inhibiting the BDNF pathway.
Yeah.
You know,
I
so,
think, yeah. I think that's an excellent suggestion and an excellent idea. You know, you're probably familiar with this phenomenon of post-ictal psychosis that you'll commonly see in patients that have flurries of seizures. And I've thought I've thought since the very first patient that I saw with that in the early 80s, I've thought those are endogenous protective mechanisms activated by the brain to reduce the likelihood of another seizure. But they're simultaneously producing this behavioral disorder. And I think, you know, hopefully we could exploit the good parts of that seizure if we could follow that, you get that cluster, bingo, then you give them the peptide for 3 days. And I think you could actually reduce, you may transform medically refractory to medically responsive. But that's why I'm asking you that, together with catastrophic epileptic encephalopathy in these infants. I think those are great instances in which seizures are beginning seizures. But I need your help in the design of a clinical trial so that we can test that hypothesis.
Then the last question is, at the threshold of translation into a human trial, are there any thoughts about the potential safety of inhibiting this pathway through the peptide?
Yes. Okay. Excellent. Another excellent question. And the answer is yes, there are. And specifically, what we would be concerned with is that while the individual is on the peptide, that there could be an impairment of acquiring memory, because we think that this signaling pathway may be contributing to our memory function. Now, we expect that would be transient, okay? Because when you take the peptide away, they should be fine. And that would be the major concern. So, and it could be that, given that a phase 1 clinical trial might be optimally done in patients with the disorder.
Okay, we just got another question here on the web. Is there any imaging biomarker PET ligand under development for BDRF or TrkB? would that be a good strategy to know how humans do in vivo and stratify them like Alzheimer's disease trials have done?
Yeah, another excellent question. And the answer is we do not have such a readout at the present time. And okay, and we're yeah, we don't have one. And but I agree. The key for us is knowing the natural history of the disorder to design a clinical trial.
Mm-hmm. True. I guess, I mean, my personal first thoughts about this. I guess that, trials may need to proceed in just individuals with seizures, with established epilepsy, just like any other anti-seizure medication, despite the specific, the strong scientific evidence of the specificity of this agent to prevent seizures after an insult.
Mm-hmm.
But again, I mean, to recruit enough numbers and assess its efficacy in a wide population, I don't know. I don't know if you've considered just a regular,
Yep.
But this brings us to, I think, 9 o'clock. Thank you very much, Professor McNamara. That was outstanding. We appreciate it. And stay safe and have a great day.
Mohammed, thank you very much for the opportunity. It was a delight, and my best to you.
Thank you.